Good afternoon, everyone, and welcome to today's PHO Microbiology presentation on what's new in Group A Streptococcus, gas, and invasive gas disease in Ontario. My name is Irene Armstrong, and I'm a public health physician at Public Health Ontario, and I have the pleasure of moderating today's session. I am joining you today from Toronto, but for thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, the Haudenosaunee, and the Mississaugas of the Credit. The land we are meeting on is covered by Treaty 13, signed by the Mississaugas of the Credit, while the province that we serve is covered by more than 40 treaties and other agreements, as well as unceded territory. Ontario has been and is still home to many diverse Indigenous people from across Turtle Island, and we are grateful to have the opportunity to work on this land. We acknowledge this as part of Public Health Ontario's and our own personal commitments to truth and reconciliation. It is now my pleasure to introduce the speakers for today's presentation, Dr. Erin Campigotto, Dr. Michelle Science, Dr. Allison McGeer, and Dr. Joseph Zeppa. Dr. Erin Campigotto is a medical microbiologist and the head of the Division of Microbiology at the Hospital for Sick Children and an assistant professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto. Dr. Michelle Science is a pediatric infectious disease physician and a medical advisor for infection prevention and control at the Hospital for Sick Children, IPAC physician at Public Health Ontario, and is an assistant professor in the Department of Pediatrics at the University of Toronto. Dr. Allison McGeer is a senior clinician scientist at Sinai Health, professor in the Department of Laboratory Medicine and Pathobiology at the University of Toronto, and coordinating investigator at the Toronto Invasive Bacterial Disease Network, also known as TIBDN. Dr. Joseph Zeppa is a clinical microbiology fellow at the University of Toronto. He completed his PhD from the University of Western Ontario and postdoctoral training at the University of Pittsburgh. I'm now going to pass it over to Joseph for the first presentation.
Thank you so much, Irene. Here's a disclaimer as well. And today's learning objectives are to describe the epidemiology of invasive and non-invasive Group A strep in children, describe recent changes in the incidence of iGAS in Toronto and the Peel region, review the incidence and epidemiology of iGAS in homeless persons, and determine the viability of whole genome sequencing to differentiate invasive from non-invasive clinical isolates. And we'll be taking these in a bit of a reverse order. So today I'm going to talk to you about a project that I was a part of while completing a rotation at the Shared Hospital Lab at Sunnybrook Health Sciences Centre in Toronto, Ontario. And we've called this Comparison of Pharyngeal and Invasive Isolates of Strep pyogenes by Whole Genome Sequencing. I personally have no conflicts of interest or disclosures to make. And a bit of an overview just for my session, I'll go over the background of Group A strep for myself and everybody giving our talks today. And then I'll go through some of the interesting findings we had from one of our recently published studies that you can see the reference for down below. So I'm sure everybody here is aware that Strep pyogenes is a gram-positive human-restricted pathogen. And what I find really fascinating about this organism is it's really capable of infecting and colonizing almost any tissue in the human body. And because of this, it's actually able to also cause a wide variety of disease manifestations, from the more superficial impetigo and pharyngitis to the more severe necrotizing fasciitis and toxic shock syndrome, and of course, a whole host of post-immune sequelae, such as rheumatic heart disease and rheumatic fever. Interestingly, up to 12% of individuals can be colonized, and this is primarily in school-age children. And this organism is responsible for over 600 million cases of pharyngitis worldwide each year, 100 million skin infections, and all of these result in over 500,000 deaths per year, which not a lot of people really take into context. And these are primarily due to post-immune sequelae and severe disease manifestations. Some of you also may be wondering how this organism is able to infect such a wide variety of tissues and cause such a wide variety of disease manifestations. And this can primarily be attributed to the large virulence factor repertoire of Group A strep. Unfortunately, we don't have a lot of time to go into all of these here, but one primary virulence factor of interest and important to today's talk is the surface M protein. Now, this is an antiphagocytic virulence factor. You can see in the top right-hand picture of this slide, kind of an artist's renditioning, showing the M protein coming attached to the cell membrane and then protruding through the peptidoglycan and the group A carbohydrate well into the extracellular space. What's also really important about this virulence factor is it has been used in typing for decades. So primarily it was used in a scheme called M serotyping, whereby you would take rabbit serum, and if it was able to precipitate out based on the M protein, a specific gas strain, it would be considered that specific M serotype, of which there were over 80 serotypes known. Then with the advent of sequencing, we moved on to sequencing certain portions of this M protein, and that's how we developed M typing. So they would sequence the first 30 codons or 90 base pairs of the mature M protein, and anything that was greater than 92% similar was considered the same M type. That resulted in over 275 unique M types, and this was then further subcategorized by including additional codons, and any changes in any of those base pair sequences that would result in a new M subtype as well. So just keep that in mind for the rest of this talk. So for my portion of the presentation, I am going to have to take you a little bit back in time. So here's some data that comes from the Ontario Ministry of Health, and it's showing the number of invasive Group A strep cases through the 2022-2023 season. So what you'll see is, again, on the y-axis, that number of invasive Group A strep cases, and on the x-axis, the months that make up this season. And what I really want you to note here is the gray line, which is the 5-year average of the pre-pandemic Group A strep season, so 2014-15 to 2018-19. And what's really striking is the 2022-2023 season saw an increase in essentially every month compared to that 5-year pre-COVID average. And so we were really fortunate that at the shared hospital labs, they were banking both non-invasive pharyngeal and invasive blood isolates of Group A strep during the timeframe indicated in the red box there. So we thought this was a great opportunity to see if we could use whole genome sequencing to determine if there was a genomic change that could account for this trend. So we again took our non-invasive throat isolates, of which there were 117, 38 invasive blood isolates, took the Group A strep, extracted DNA, and performed whole genome sequencing using the Illumina mini-seq, and then performed a number of analyses that I'll go through now. So this first chart here shows the absolute number and the percent of parents of our invasive and non-invasive isolates and their M type and M subtype distributions. So the data is a little bit small here. So really the take-home message that I want to show you is that we saw pretty striking differences in our M type distribution between these two different subsets. So our invasives were primarily made up of M12, M49, and M1, and our non-invasives were primarily M12 at about 70%. And for those of you who might be interested to see how this maps onto that Ministry of Health data, they also provide this information. And so our top 3 M subtypes for invasive were essentially the exact same as the 3 from this data set as well, although the percentages were a little bit different. But interestingly enough, we still had the same 3 top subtypes. So that showed that our data set was probably a decent representation of the larger subset. So we then decided to determine where our isolates fit in context of kind of the known Group A streptococcal distribution. So we took our isolates, which are shown here in this minimum spanning tree, along with over 2,000 other isolates from the NCBI database. Our isolates are highlighted by the larger circles, with the invasives being the orange color and the non-invasives being the lighter purple color. So what you can see here is our isolates cluster nicely together based on their M type. But you can see within M types, you see a bit of a smattering of the non-invasive and invasive overlapping, indicating that this probably isn't the best way to look at our isolates in order to kind of parse out the non-invasive versus invasive. We then decided to just take our isolates from this project and do a core genome phylogeny. And again, we see this nice clustering based off of M type, but we see both of our non-invasive and our invasive types interspersing amongst one another. So this, again, also isn't really helping us parse out what is unique between these two subsets. I personally am very interested in Group A streptococcal virulence factors and adhesion factors. So we then decided to do an analysis whereby we determine if there is a difference in the number of these genes in the genome. So we took about 59 different genes that are associated and known as either virulence factors or adherent/binding proteins. And we decided to see if there was a difference in the mean number of these genes within the non-invasive or invasive isolates. And so when we looked at kind of the larger categories of virulence factors or adhesion, we saw no difference between these groups. But once we started kind of diving deeper into some of these subcategories, we did notice, excuse me, that our non-invasive isolates did encode for more superantigen and DNase genes compared to our invasive isolates. We then were interested in just doing a one-to-one gene comparison between these two groups. And you can see here highlighted in green and red the differences between these two. So the green indicate the genes that were more prominent in our invasive isolates. And these included some M-like proteins, the superantigen SPA, another virulence factor known as IDES or MAC, which is a molecule that degrades antibodies, and a couple specific adhesins. And in our non-invasive isolates, the more prominent genes were the superantigen SPC, SSA, SMES, the DNase SPD1, the hyaluronidase HYLP gene, ENDO-S, which also modifies antibodies so they can no longer function properly, and the GRAB protein, as well as two unique different adhesins as well. We also didn't just want to look at genomic differences, so we decided to do a couple rudimentary phenotypic assays as well. So we took each of our isolates and plated them either on blood agar or milk agar to look at their hemolytic and proteolytic factors by measuring the zone of clearance on both of these agars. And very interestingly enough, our non-invasive had larger zones of proteolysis and hemolysis compared to our invasive, indicating that they produce more hemolytic and proteolytic factors, at least in the conditions tested. For those of you who are well aware of the Group A streptococcal literature, there is a key two-component system called control of virulence, SNR, or known as COV-RS. And so it is known that when there is a mutation in either one of these components, the organism is known to flip to a hypervirulent phenotype. And so what was interesting here is that we noted that 7 of our 38 invasive isolates, or about 18%, had mutations in this key two-component system, whereas 0 of 117 non-invasive isolates had any sort of impact on COV-RS. So this was kind of a result that was to be expected, but still interesting nonetheless. We also looked at different antimicrobial resistance genes in our clinical isolates. So these were genes that are known to encode for factors that provide resistance to things like aminoglycosides, macrolides, and tetracyclines. We overall didn't see too many genes, and there was actually no difference in any of them between our two groups. But I just wanted to highlight this data here. And so just kind of to highlight that here, what we did notice in our study was that both invasive and non-invasive isolates were represented across a diverse set of lineages, with our invasives being primarily M12, M49, and M1, and our non-invasive being M12. We did see a different prevalence in superantigen DNases and single virulence factors between our invasive and our non-invasive. It was the non-invasive that produced more lytic and proteolytic factors, and it was only invasive isolates that had mutations in the COV-RS gene. So I do have some future directions highlighted here because this is a topic I'm highly interested in, and I do realize there was a number of limitations to this study. So this was a relatively small sample size over a relatively small time frame. So I would love to expand the sample population to increase our numbers and strengthen our analyses. I think we only had bacterial data here, so it would be really interesting to gather some patient information to integrate host factors into our overall findings. Again, we had those relatively rudimentary phenotypic assays, so I think it would be really interesting to assess virulence factors, potentially adhesion and transcription factors using additional assays. And the the AST data that I showed you there was purely genotypic, and I would love to integrate some phenotypic testing of these isolates as well. And so I'd like to acknowledge the team that I worked with here, a friend and colleague, Ellen Avery, who started this project out, who is also just graduated and passed her clinical microbiology exam. So congratulations, Ellen. And a great team at the shared hospital lab that operates out of Sunnybrook. Everybody here was fantastic. And this was primarily driven by Dr. Rob Kozak and Finn McGuire. So thank you very much to everybody there. Thank you so much to listening. And I will pass the mic off now to Aaron Campigotto.
Thank you, Joe. So first of all, I'm going to present a complementary study on Group A strep in children and comparison of invasive and non-invasive isolates. I will be presenting today on behalf of myself and Dr. Michelle Science, and we have no conflicts of interest to declare related to this work. So our learning objectives for this portion will be to really look at the epidemiology of invasive and non-invasive Group A strep isolates, similar to what was just presented, but we'll focus on children and what we saw here at the Hospital for Sick Kids. We'll look at both the clinical presentation of the invasive and non-invasive isolates that we reviewed and primarily focus on the M-type distribution of these different isolates based on the clinical presentation. So just some background and potential confounders to take and consider as you look at this data. One challenge that we have, especially when reviewing some of the non-invasive literature and results, would be to look at the potential for colonization rather than an infection associated with Group A strep, particularly in children. We know that within children, the oral pharynx may be colonized in up to 20% of children, and you can see that in this study on the left-hand side, where younger children in that 3 to 5 year age range have highest rates of carriage potentially. And so any samples that we have where the pharynx was sampled, we do have to put this into context of the clinical picture. The other piece that we do have to adjust for and consider is seasonality. So colonization as well as clinical disease that we'll see does follow a kind of typical seasonality picture. And so lower rates of colonization, usually lower rates of pharyngitis and less invasive disease in the summer months as compared to the winter. So we want to look longitudinally across the time period as well. When we look at prevalence studies for non-invasive Group A strep disease in children, unfortunately, there's not a lot of literature out there. The studies are challenging to really capture all the clinical pictures and clinical syndromes that Group A strep may cause. They are often very common presentations as well. So to actually capture all the data is challenging. Those clinical presentations that we usually do see are really pharyngitis is the #1, but even other skin and soft tissue infections, scarlet fever, and others. And you'll see within our data here, we do capture all of those different types of non-invasive clinical disease. When we look at the seasonality picture, so on the right-hand side, this was a study in children in Spain. The focus was really to see how COVID affected invasive and non-invasive Group A strep disease, but I think it really shows that the seasonality picture continues and that invasive and non-invasive disease follow the same sort of trends, and that both during COVID pandemic, there was a decrease in both non-invasive and invasive disease, but since 2022, that has reversed. And then finally, taking a look at our Ontario data. So this is our Ontario data showing invasive Group A strep rates for the 2022 to 2024 period. Two things I wanted to point out on this graph is one, as Joe also described in the overall data, we did see that increase in invasive Group A strep starting in 2022 and then continuing into 2023. And then for the study in the timeline of the invasive and non-invasive isolates that we are presenting here, you can see how we overlap this period. So the kind of light purple colored mauve line shows the 2022-2023 less than 18 invasive Group A strep isolates, and the period that we start for this study that I will show really started a few months prior to the increased invasive group A strep. We continued to study all isolates that we received here at Sick Kids until almost the end of this graph in the 2023 to 2024 period, but you can see that we captured about 1 to 2 months at baseline prior, and then another 2 to 3 months of really return to baseline following that increase in group A strep, invasive group A strep disease. So the study that we'll present for the next few slides and the rest of our presentation here will be based on group A strep isolated at Sick Kids between December 1st, 2022 to August 31st, 2024. And as you saw in the previous slide, that time period was chosen to overlap with the increased invasive group A strep that we saw with some return to baseline on either side. Within our study period, we sampled 1 specimen per patient per 2-week period to be included in analysis. So a single patient on a few instances could contribute more than 1 isolate, but we considered a separate event. Again, very few patients fell into that group. And then what we wanted to look at was clinical characteristics and bacterial characteristics. So from the clinical characteristics, we'll present age, collection site, clinical presentation primarily. And we do have other variables that will not be presented. And then from the isolates, so the bacteria, we also performed whole genome sequencing. This was performed using Oxford Nanopore technologies. And the focus of this talk will really look at the M type that was obtained. So when we look at the age distribution of patients with invasive and non-invasive group A strep, you can see that overall we had 408 patients enrolled in this study. And that breakdown on the right-hand side shows the non-invasive versus invasive group A strep. So 319 in non-invasive group A strep and 89 invasive. There was a slightly lower median age in the and interquartile range for the invasive group A strep disease. Part of this may be driven, as you can see, a lot of the non-invasive group A strep really cluster between the ages of 4 to 10 or 11, maybe 12 years of age, which would correspond with a lot of the pharyngitis cultures. We know that group A strep pharyngitis rarely occurs in children 3 and under or children under 3. So those sorts of cultures would unlikely make up the proportion or high number of proportion in that age group. So it does fit with the clinical pictures that we see from group A strep or non-invasive group A strep disease. When we look at the different characteristics of the patients, again, from their underlying medical conditions and male sex, so you can see, again, fairly even distribution between non-invasive and invasive group A strep between sex. And then in terms of underlying medical condition, well, we don't have any statistical analysis, just looking at the all rates in terms of a skin condition and any predisposing factors for invasion potentially, or underlying respiratory conditions, which again may predispose to potentially more severe illness due to group A strep, or immunocompromised due to malignancy transplant. The non-invasive and invasive groups were relatively similar. The specific indication for the test or for the group A strep result is shown here. So you can see the typical clinical presentations for on the left-hand side, the non-invasive group A strep isolates, and on the right, the invasive group A strep isolates. So from the non-invasive isolates, the most common presentation, not surprisingly, was pharyngitis, made up of this green section of the pie chart, followed by cellulitis, and then other skin and soft tissue infections that were not cellulitis. Of the total number of non-invasive group A strep isolates, the rest of the data really focuses on those who have also M type. So WGS, whole genome sequencing, was completed on 81 of those 88 isolates successfully. When we look at the invasive group A strep presentation, again, the most common was skin and soft tissue infections, often necrotitis and fasciitis, followed by pneumonia in a large portion as well, and then bacteremia. That other group makes up a small number of cases where we would have, say, meningitis or other invasive group A strep presentation. When we look at the clinical presentation, again, broken down by age group, you can see that the green bar in the left-hand side represents pharyngitis, and again, not surprisingly, the highest number of non-invasive isolates was in that 4 to 10-year age range, and it was primarily due to the pharyngitis isolates. You can see under 3 years of age and over 12, there are very, very few isolates that belong to that group. The other clinical manifestations were relatively constant among the age ranges. And finally, in terms of the invasive group A strep clinical presentation, it really, there's not a lot that really differed among age groups. There was a large proportion of bacteremia as well as pneumonia in the under 5, particularly the under 5 age grade. When we look at the M types based on the clinical presentation of invasive versus non-invasive, so the blue lines being the non-invasive isolates, the red, the invasive isolates, just looking at absolute numbers. So when we look, the most common M type among both the invasive and non-invasive isolates was the M1 UK. You can see that approximately there were 2 times as many non-invasive M1 UK isolates as M1 UK invasive isolates. This was followed by M12, but you can see there was many more non-invasive M12 isolates compared to M12 invasive isolates. And then the really difference here is in the next few subtypes. So M4 and M2, where they were the next most predominant non-invasive, but we saw very few invasive cases, and M1, where we saw more invasive cases, especially more invasive cases compared to the prevalence that we saw among the non-invasive isolates. When we just look to see if there is a difference of M-type distribution by age, really, this is an absolute number. It doesn't change if we look at these graphs by proportion, but really nothing jumps out. There's no significant findings. Primarily the increase in the 3 to 10-year age range for M1 UK that is seen is again, it was the most common M type observed in our non-invasive isolates, and really among our patients with pharyngitis. And then when we look by clinical presentation, this really just shows what we were talking about there. So this is a percentage of M type by the clinical presentation. You can see that for the most part, the 3 key differences I think were there's a decrease in the M1 as a proportion of cases by pharyngitis, and we saw that M1 actually had more invasive disease among isolates observed. The M12, we saw slightly more patients with, or less patients with pneumonia, and then M1 UK, we saw slightly more patients with pneumonia. And so finally, that really comes up to our conclusions is really that it was M1 UK and M12 had similar proportions to their invasive and non-invasive isolates, but M1 UK predominated among both clinical cohorts. Our M1 isolates particularly were more prevalent among our invasive isolates and had very few non-invasive, particularly among our pharyngitis isolates, patients with pharyngitis. And then M2 and M4 were rarely seen in invasive disease, but were fairly common among our non-invasive isolates, particularly in pharyngitis. And with that, I can pass this over to Dr. McGeer.
Thanks very much, Erin. Good afternoon, everyone. And I'm going to close this out talking a little bit about what we've been learning about iGAS within TIVDN surveillance. TIVDN, as you know, does population-based surveillance for invasive group A strep and other diseases in Toronto and Peel region, and probably includes a large number of other people who are on the call, and certainly all the panelists. So first, just a quick review of what we've seen with pediatric invasive group A strep disease. This analysis done by Halima Dabaya Yunus, who was an IPAC fellow for Jenny Johnson here at Sinai for a couple of years, and went back to the beginning of TIVDN and iGAS surveillance in 1992. And what you can see on this slide is iGAS split up. So the line with the dots on it is everything other than respiratory infections due to iGAS. And you can see that it's a perfectly flat line from 1992 to 2019. That's interestingly, despite the fact that iGAS complicating chickenpox, of course, completely disappeared, used to cause 16% of cases, and it disappeared. Despite that, iGAS predominantly soft tissue infections have remained stable. And then in the bars, you can see respiratory tract infections, and you can see that there was an increase in respiratory tract infections that started in the mid-2000s and increased, and then, of course, decreased during the pandemic and rebounded really strongly after the pandemic. So pediatric iGAS has been increasing for about a decade now, mostly because of an increase in respiratory tract infections, which are only about a quarter of disease. The second thing to know about what happened during the pandemic is in this EMM typing graph, and all I want you to look at is these light blue bars at the bottom. These are EMM1, includes both the old EMM1 and the new EMM1 UK. But if you look at 2020, 2021, and 2022, what you see is that EMM1 is the one that just disappeared. Everything else looks much the same during the pandemic. There's a little bit of a decline, but most of the pandemic effect was in fact a decrease in EMM1 during the pandemic. And then it came roaring back, as did EMM12, right? So if you look at EMM12, you can see it's been there all the time, but there was a lot of it in 2023. In contrast, some of the other common EM types, 3 and 4, just kind of keep going along, and they don't change very much immediately after the pandemic. Interestingly, some other places in the world got this resurgence before us, and in the last 9 or 12 months, they've now seen a resurgence of EMM3. You can see EMM3 may be appearing at the end of 2024, so that may be in the cards for us coming later. It's really hard to tell. The second thing just to know about these EMM types is that the most advanced Group A strep vaccine is a 30-valent vaccine, mostly with M protein antigens on it. And in pediatric disease, we cover nearly 93% of the strains, and hasn't changed over the last 30 years. So as long as that vaccine keeps going, we'll be doing fine when it comes. Okay, now it's not only pediatric disease that has increased. This is now incidence of iGAS in adults aged 15 to 64. And you can see that although EMM1 is a lower percentage, there's also a resurgence of EMM1 and 12 in 2023 in younger adults. But there's also this increase that started in 2012-13. It's a little harder to tell now whether we're plateauing in adults aged 15 to 64 or whether that increase is actually continuing. And finally, here's the data on older adults over the age of 65. Again, you see an increase, although it's not as dramatic, and it looks as if there's an ongoing increase in 2024, 2025. So this is very similar to what's been seen in the United States, just reported earlier this year. In kids, about a 30% increase, but in adults, almost a doubling of the increase of invasive group A strep from before 2010 to the years after the pandemic. Okay. In the last few minutes, let's talk a little bit about what we know about Group A strep in housed and houseless people. As we all know, homelessness is an increasing problem across much of the world, and certainly in Toronto. And one of the things that is increasingly evident as our population of homeless people, almost all adults, increases, is that there are really sizable increase in risk of a variety of infectious diseases, and that includes invasive group A strep. So here's the rate in overall adults in 2022 and 2023 was 3.5 per 100,000 in housed adults and 103 in homeless adults, and 7.2 and 220 in houseless adults. We were expecting that because the resurgence was primarily AMM1 and would be mostly in children in 2022, that we might not see as big a resurgence in disease in homeless adults as we saw in housed adults. But in fact, as you can see, the ratio is almost identical. So about a 30-fold increase in risk in houseless adults in both years. And then if you look at the characteristics in housed and houseless adults, you see what you would expect. Average age of homeless adults with Group A strep is significantly younger, because the population is significantly younger. They're less likely to have immunocompromising conditions and to have autoimmune conditions than housed adults. They're more likely to be current smokers, more likely to use intravenous drugs. And when you look at other risk factors, they're significantly more likely to have non-intact skin, obviously a condition of the challenges of living in an unhoused situation. When you look at their clinical diagnosis and outcomes, they're significantly more likely than other adults to have a soft tissue infection and less likely to have bacteremia without focus and upper respiratory infections. Interestingly, not a significant difference in pneumonia, but that there is a trend. And this is really about the shift to skin and soft tissue infections in homeless adults because of the challenges of maintaining skin condition without a home. You can also see that their risk for osteomyelitis is higher and their risk for endocarditis is dramatically higher. That risk is not actually being houseless. That risk is IV drug use. And when you separate out the IV drug use and the homelessness, it's the IV drug use, obviously, that is really the risk factor behind infective endocarditis. The other thing you can see is that homeless adults are less likely to have streptococcal toxic shock syndrome and significantly less likely to die of their invasive group A strep. I suppose some small silver lining to the dramatic increased risk. And if you look on this next slide at EMM distribution, you'll see likely why. So the EMM distribution in housed adults looks much more like children. So EMM 12, EMM 1, and EMM 89 more common in housed adults than in homeless adults, and more commonly in homeless adults, EMM 49, 82, 92, 74, and 80. And of course, EMM 1 is the most virulent in general, particularly EMM 1 UK, of the Group A strep strains. So it's not surprising that you see fewer infections in less mortality, sorry, in homeless adults than in housed adults. And also that likely explains respiratory tract infection. EMM1 is associated with more severe infection. It's also associated with a greater likelihood of pneumonia and respiratory tract infection than other EMM strains. So in conclusion, Most of the pandemic-associated increase in iGAS was associated with reduced transmission of EMM1, which is persistently more common in children than adults and also more virulent. This is really dramatically different from pneumococcal disease, where what we saw was an overall decrease in every serotype of pneumococcal disease during the pandemic. So although these are both streptococci and they have some similarities, very different behavior in response to public health restrictions during the pandemic. Incidence of iGAS appears to be increasing, so it's a very good thing that we are looking at the possibility of vaccines in the relatively short term. Homeless adults are more than 30 times more likely to develop iGAS infections. There's probably a limited amount that we can do to reduce that risk. It's, you know, the simple answer is people need housing. And it's unlikely that we can do much to improve people's skin condition and reduce risk. We can likely reduce risk, perhaps a little bit more than we're doing if we can maintain safe injection sites over time. We'll see how that goes. And finally, I showed you the data on coverage for iGAS in children. Overall, it's a little bit lower in adults, so it's about 75% of all strains would be covered by that iGAS vaccine. I'm going to stop there so that we have some time for questions for everyone. Thanks.
So thank you, Dr. Erin Campigotto, Michelle Science, Allison McGeer, and Joseph Zeppa for those fascinating and insightful presentations. We're now going to move to the Q&A segment to answer questions from our live audience. And so a quick reminder to everyone, if you haven't done so already, please enter your questions into the Q&A pod. Okay. Okay, so the first question we have here is someone said, I joined late, so apologize if this is already covered, but can you please explain why iGAS cases have increased post-pandemic? And I think that's really, maybe I'll turn that one over to Allison. I've heard a lot of theorizing in general from people about this.
Sure, well, I can start. So I think the first answer is that, as with many other pathogens, we had a reduction in transmission of particularly EMM1, Group A strep, but probably EMM12 that we just didn't see because it's more of a colonizer during the pandemic. And we then built up 3 cohorts of young children who didn't have acquired immunity to, influenza, RSV, Group A strep, strep pneumo, and as a consequence, we saw a resurgence. Again, there's undoubtedly things to learn if we knew the questions to ask about why the resurgence happened at particular times and why it was different in different timing in different areas, and why we really haven't seen much of a rebound in strep pneumo, but we saw a big rebound in Group A strep and in flu and RSV. So the short answer is it's unquestionably associated with reduced transmission during the pandemic. But the long answer is it's actually really complicated and there's a lot of things we don't understand.
Thanks, Allison. I don't know if anyone else wanted to add some thoughts to that as well. So maybe I have if no one else wants to respond, I have a sort of a hot pursuit to that one. So Allison, you mentioned that like M1 went down like quite a lot during the pandemic, but the other M types sort of stayed along at the same rate. And you did make a reference into your response now that about some of the things maybe being colonizers, but can you explain why you think that there was that reduction in M1 and not in the other M types as much or at all?
No idea. Okay. I mean, I think it's it's clear that EMM1 causes a lot of disease in children, more disease in children than adults. And so, I think we tend to think of strep pneumo and Group A strep as uniform things, right? But the truth is that they're not uniform. And when you, with the wisdom of hindsight, looking at what happened during the pandemic and the distribution of EMM1 over time, I think it's likely that children are the big reservoir for EMM1. It's not nearly as clear that they're the big reservoir for all of those ones with higher MM types that you see more commonly in homeless adults. And exactly why that should be true, I'm not sure, but I think the differential is likely that the reduction between kids for EMM1 was really important, but we didn't have as big an impact in whatever other reservoirs there are for Group A strep out there. Not a very satisfactory answer either.
Thank you for that, though. I had a question for Joseph, and I'm wondering, I'm not a medical microbiologist, but you were when you were going through your slides, you were talking about you know, how non-invasive gas exhibited more proteolytic and lytic factors. And as a non-microbiologist, I would have thought it would have been invasive gas that might have exhibited more of those. And I wondered if you could explain a little bit about that.
Yeah, of course.
And what
I think
the meaning is. Yeah.
Yeah. I mean, I think it seems like a bit of a counterintuitive finding until you kind of think about that these organisms probably, you know, maybe I'm personifying a little bit too much, but I don't think they really want to cause a lot of these invasive diseases. It's not really how they spread. They would spread by respiratory route if they're pharyngeal colonizers, or skin-to-skin contact if they were more common to cause skin manifestations. And so I think a lot of these virulence factors also co-evolved with their capacity to cause, you know, either asymptomatic colonization or some of the more less severe manifestations. So the fact that maybe, although once they get into a sterile site like the blood, these things can cause, these virulence factors are very well associated, you know, with superangens causing toxic shock syndrome, streptokinase, you know, and other factors, you know, getting through some of our tissues. But I think we shouldn't forget that these factors are also very important for these organisms' ability to, you know, colonize asymptomatically or otherwise our, you know, respiratory mucosa. And so I think it also does make sense that they might produce more because, you know, they're probably competing with a lot more organisms in those microbial niches compared to once they get into the blood, they're probably the only thing in there in most cases, and they can kind of run rampant. But that's probably my best thought as to why they might produce more of these factors in those environments.
Oh, thank you for explaining that. I also had a question for Aaron and Michelle, and I'm just wondering, why do you think that gas pharyngitis is not seen in children less than 3 very commonly? Or does one actually know? You made a reference to that in your presentation.
I'm not sure if Michelle knows. Like it's very well described. It's part of the clinical criteria for treatment and even testing. And the rationale behind, I'm not sure myself of the clinical basis for that though. I'm not sure if Michelle does.
I think Michelle's coming on.
No, I'm sorry, my internet keeps cutting in and out.
No.
I think it's one of those things, and Allison, please feel free, or Joseph, if you know. I think it's one of those, as you say, it's well described. There's some sort of thought that, you know, immune system development, exposure and transmission in sort of the younger children, they're not of school age or necessarily in daycare. So I think it's probably multifactorial related to the immune system and exposure risk.
Oh, thank you for that. I just had one last question. I don't know if there's anyone else, if anyone wants to add a question to the Q&A chat, I would love to present that to our esteemed group of panelists. But I often get asked about invasive group A strep, and people will say, well, there's this M type, what's associated with it? There's that M type and what it's associated with. And has anyone ever sort of developed a catalog of M types and correlated with clinical presentations? And I wonder if that would even be stable across different areas, or do different M types always tend to have the same clinical presentations?
I can take a first stab at it. So yeah, I think there is pretty good association with M type and different disease manifestations. And we didn't really cover it here to a great extent, but there's other similar but there are distinct aspects to it. M typing, such as M patterns, which kind of broadens the genes that you look at, and kind of as you increase the number of genes you look at in your typing schemes, you can definitely start associating fairly well with M pattern types, is what they're called. And there was a slight allusion to them on my one slide because I like how it categorizes both the M typing and the M pattern typing. But anyways, there is association with those M pattern types and their ability to primarily like to infect the skin, the pharynx, and then there's an M pattern type as well that's a bit of a multi-factor player. So you're almost equally likely to see it in the pharynx or on the skin.
Thank you. And is there a catalog people could look at for inquiring minds who want to know?
There's some great papers. Deb Besson, I unfortunately forget her affiliation, but she's got some really good papers that do have good distributions of the different M pattern types. And I'm sure there's others if one of the other panelists knows, but that's always the ones that come to mind for me.
Well, thank you very much. I see no more questions in the chat, so I think we're going to come to a wrap up now. And as we wrap up today's PHO Microbiology Round session, I would like to sincerely thank Dr. Erin Campigotto, Dr. Michelle Science, Dr. Allison McGeer, and Dr. Joseph Zeppa for presenting. I would also like to thank everyone who joined us for today's event. You can expect to receive a brief and anonymous survey for today's session, so please try to complete this to help us in improving our programming. Thank you so much, and wishing everybody a wonderful day.